and then you can go inside. So that's how they stay alive in the winter. And you can imagine if it's 20 below one day and 15 above the next day and 10 below the next day, that it's more work for the bees to maintain their cluster and keep warm and not use up all their food than if they were at a constant temperature. So that's the logic behind putting bees indoor in storage at about 40 degrees Fahrenheit, where it's kind of constant, and they at that temperature they use the minimum amount of resources. So added to that is the realization from some previous work that Varroa mites, these parasitic mites that are devastating to honeybees, actually cannot withstand CO2 levels as high as the honeybees can. So Brandon and his students have been working toward developing a system where honeybees can be put in cold storage and maybe storage with metabolic gas capacity to control it. So in other words, CO2 storage, they can raise the CO2 levels and to a point that it's more that is harmful to the mites and won't hurt the bees. So that's kind of the goal of that research. And they've been making some really nice progress. We have some refrigerated gas controlled containers over at Othello. So our research at that facility in the last 6 months has really been limited to using those storage units. And I think the last experiment that they've just finished, is banking queens. So queen producers in California, in almost an opposite situation, they can make queens for sale, tens of thousands of queens, hundreds of thousands actually, all the way up until, let's just say, about July. But then they have a demand for more queens from customers in September. So one idea has been to try to bank the queens in cold storage. So currently the beekeepers put all the queens inside cages, inside large colonies, and they lose about 20% of them over those 2 months while they're waiting for the customers to order more queens. Because in September they can't make queens, but they can sell the queens they made earlier. And so this experiment involved, I believe, about 18,000 queens being put in. Maybe it was 1,800. I don't want to be that far off. It was a lot of queens. I think it was 1,800 in these facilities. And Brandon just last week carried the queens back down to California to be compared to the queens that were stored outside. So anyway, the indoor storage and brood break is a pretty active area of research that's coming out of that facility. We have historically given queen breeding and keeping workshops at WSU in Pullman, and we planned our next ones at the facility at Othello. And because of COVID-19, as you might guess, that's deferred. We're not doing that. One long-term project that we've been doing really since I got here has been queen breeding. And for about the last decade or so, we've actually been bringing in semen from old world populations of honeybees. So honeybees are not native to the world. They're native to Europe, Africa, Central Asia, the species that we have here, Apis mellifera. And so if you want to breed those bees or increase genetic diversity, you need to get breeding stock from the old world. We're not allowed to bring in bees from the old world, but we are allowed under permit to WSU to bring in bee semen. And Brandon, as part of his PhD work, actually developed the means how to freeze that in liquid nitrogen, just like we do with cows and pigs and humans. We were unable to do that with bees until he made sort of a breakthrough. So now at WSU, we have a genetic repository with semen from a number of subspecies around the world. And as far as I know, we're the only facility in the US doing that. We do deposit honeybee germplasm, the semen, into the National Animal Germplasm Facility in Fort Collins, Colorado. It's run by the USDA. But no one else that I know of is using cryo-preserved semen in a breeding program. What's going on here? Okay, there we go. Okay, so that brings us up to program bees. This is our standard breeding program that we've been doing since about 2000. We got a USDA grant along with Cornell University, and for more than 20 years, we've been selectively breeding honeybees and providing them to beekeepers in the queen production industry. And along the lines of this reproductive biology, I told you that Brandon, as a graduate student, perfected the means to freeze semen and make it usable. He now has a graduate student, Connor Off, who's been working on ways to further improve that. And I have a student, Kirsten, who's also working in that area of reproductive biology and trying to understand, I'll just say what goes on in the female reproductive tract for honeybees, because as you may not know, honeybee queens fly up in the air as virgins and mate with about 10 to 20 males on her nuptial flight. And then they come back, and it's sort of a mechanism to promote outcrossing to reduce the effects of inbreeding, because honeybees are very sensitive to inbreeding. In other words, if they breed with closely related males, it causes them to have eggs that are not viable. I'll just put it that way. Yeah, so that's going on. And then the one other major direction of the lab the last several years has been using fungi to help honeybees, honeybee health. And there's a kind of a two-tiered process or a two-pronged approach. One of them is using fungi to directly attack and kill the mites. The Varroa mite is a parasitic mite, lives on honeybees, and we can put a fungus in the beehive that kills those mites. And that work's been done by Nick Nager and Jennifer Hahn. And Jennifer has been selecting this fungus. The fungus was known, but it's normally a soil-dwelling fungus. It does not do well in honeybees, beehives, because of the higher temperature. And she's been selecting for fungus that can withstand higher temperature and is more viable in the honeybee colony as far as being more infective. So she's been selecting for virulence in this fungus, and we're at the point now where we're growing up large quantities. By that I mean large enough quantities to do full-scale outdoor tests with hundreds of colonies. So we're pretty excited by that. The other work that we've been doing has been using fungal extracts to reduce viruses in honeybees. So it's believed that one of the ways that these mites are really very harmful to honeybees is they transmit and amplify bee viruses. And those viruses, and I guess now with the COVID-19, we can kind of understand about infectivity. And so I'll talk about that a little bit, but basically there are these polypore fungi that live on trees, and these fungi live with the tree, and you might think of it a little bit as a tit for tat kind of thing. The fungus prevents viruses and other fungi from attacking the tree. So they're beneficial in that sense to the tree. But the deal they make with the tree is that when you die, we will consume you and make a lot more copies of our fungus. So these are polypore mushrooms. You've seen them if you've been out in the woods and you see these shelf mushrooms and things growing on a tree. A lot of those species contain antiviral compounds themselves, and in fact, they're even used, in human medicine. And the update on the giant hornet. So that's where we're going to go. Sorry for my long-winded introduction. So let's just start with the giant hornet because it's probably the more exciting thing for a lot of folks. This is Vesta mandarina. It's a huge hornet from East Asia, and my first introduction to it was in Korea, where it was devastating apiaries around South Korea. And the main control agent that they had, tool, was to hire school children to kill the hornets with badminton rackets. So you would go to apiaries and there'd be broken badminton rackets all around the ground. These hornets are social. They're a social insect, so they can recruit sisters to come and attack the apiary, and they're kind of specialized on honey, so they can destroy a honeybee colony certainly in less than a day. And they go in there, they eat the individuals, they grab them, fly up to a branch, peel them, eat the flesh from their thorax, and then grab another one. And once they get loaded up, they go back to the hive, to their nest. They tell their sisters, hey, there's honeybees over here. And then everybody goes over there and they just wipe it out. Once they destroy most of the field force of the honeybees, they then move into the hive and defend it. So if you're a beekeeper and you go up to your hive, with your short pants on and your smoker, thinking you're going to just check out your nice bees, they'll then come out and sting you. And they have about a 1/4 inch long or 1 centimeter long stinger and can do some damage. I believe they do kill a number of people in Southeast Asia. So there, as you can see, the size of it compared to a honeybee, and they are you say, well, then why do we have honeybees in Japan, for example? There's a honeybee, Apis cerana japonica, that lives in Japan. It's related to our Western honeybee. The honeybee that we have here in Washington State is Apis mellifera, and it's the European honeybee or the Western honeybee, whereas in Japan they have Apis cerana, the Eastern honeybee. The Eastern honeybee, even though it's smaller, and in some ways you might think it being kind of a wimpier bee, they have a defense against this hornet. And that defense is to glom onto it and shiver. Remember, they can shiver their flight muscles and make heat, and then they kill the hornet with heat. So you can see there's a hornet surrounded by Apis cerana, and they are cooking it with their flight muscles. Unfortunately, that knowledge hasn't been passed on to Apis mellifera. So Apis mellifera is just a dead duck for these hornets right now. Okay, so the story for us in Washington State or here in the US, this is, there are a number of things on the horizon that we really don't want to be here, and this would have been high on the list. But in Nanaimo, it was first found in Canada, and then a couple of specimens were found in Washington State in 2019. And the WSDA decided to make a major effort to try to delineate the locations of these wasps and try to eradicate them. And I believe Canada is doing a similar effort. So they've put traps out. They've notified beekeepers. Of course, beekeepers, if you think of it, could be your first line of defense because they are, they're looking at their hives and they would notice their hives being destroyed by a bunch of hornets. I can tell you that when the call was first put out, as you might imagine, there were so many calls of people that saw yellow jackets on their picnic table and called it in and things like that. But, so in 2020, apparently there's been 5 sightings or finds here in the state, and they found 1 queen, a virgin queen, and apparently not mated, and 1 male. So if you guys don't know the life cycle of yellow jackets or even bumblebees or these hornets, it's that they are typically a seasonal colony. So in the spring, mated females, i.e. a queen, each start a new colony by themselves. So they overwinter under a piece of bark or something, rock, and then they come out and they forage on their own. So in this case, the queen hornet would be catching, caterpillars or insects or honeybees, whatever insect prey they can find, and they would raise an initial batch of workers. And once they have a few workers, maybe a dozen or less, they then stop leaving the nest because, of course, if they were to die, that would be the end of the nest. So once you can think of them as being kind of really valuable for the colony. So once they have workers, the queens stop leaving. The workers then do all the foraging, and they build up to maybe dozens, hundreds of workers. I'm not exactly sure with this species. Usually it's hundreds, maybe low thousands. And then as the season begins to end, later in the summer, they switch over, and instead of making workers, the queen makes males and females. So all the workers, of course, are females, but they're not, sexually complete. They can't really mate and reproduce. So, and that has to do with sort of the way they're fed and some other things. But basically, into the season, they make a bunch of males and females. Mating happens, and then everybody dies except the mated females, who are then effectively the queens for the next generation. So this year, so far, in Washington State, they found 1 uninseminated female and 1 male. So that just symbolizes that we're near the end of their season, and they've caught some dead workers. In the areas where they found these, they've moved honeybee colonies into those areas to try and capture some live workers. The idea being, if they have the live workers, they can use that to try to locate the nest of the wasp. So no success in that yet, but the fact that we found males and females suggests that there's at least 1 colony out. So I believe the state is kind of determined, and certainly beekeepers are doing their best to help to try to find out what's going on with this. And if you want to know more, there's guidelines put out by the USDA at this site, and you can learn more about these hornets. A colleague of mine, Justin Schmidt, you may have heard of him, the Schmidt Sting Index. He's a retired USDA bee specialist who, as a hobby, has stung himself throughout his life with every stinging hymenopteron he can find. And I will tell you that he says this is one of the worst, and he's stung himself with a lot of different things. Okay. Are you guys still there? I guess so. I feel like I'm...
Yep, we're still here.
Okay, good. I'm sorry.
You're doing good. I haven't seen any questions.
What if I'm talking to my computer?
Oh, you're good.
Thanks. Okay. Yeah. Okay, so there's that. So I'll start now with I'm gonna go over kind of two things that we're doing now, if it's okay with you. One of them will be our breeding program a little bit because it's cool and it's the stuff I really like. No, I like it all. And then the second will be the work with the fungi. Okay, so you already know because of the intro that honeybees are not from the New World, right? They are all from the Old World, and they were introduced by European colonists. And in 1922, we passed a law restricting the further importation of honeybees. And it's because of a different mite that was found in Europe but had not been found in the US. So starting in 1922 and continuing until now, 98 years later, you cannot bring honeybees into the US. But we were allowed, this is old history, about 15 years ago, through a bunch of letter writing and the fact that the US was importing some honeybees from Australia. The mite wasn't in Australia at that time, and so the APHIS allowed some importations of that genetic material. When I say that there's the U.S. Honeybee Act restricted further importation of honeybees, that is true, but it reserved to the Secretary of Agriculture the right to bring in new genetic material. And so the USDA, basically some laws were changed that for a few years allowed the importation of material from Australia. And if you have questions about that, I can tell you it's really a much more complicated story. But we did use that as an opening to ask APHIS for a permit to bring in genetic material from the Old World, the original sources of honeybees, in the form of semen to use for breeding. And we got that permit. We have a quarantine apiary, and all the semen is screened by the USDA for viruses, and only is the genetic material allowed to be released. So that's number one on that little list. Number two, WSU program stock. Those are just bees that we have been breeding for about 20 years from the original genetic material available in the US. So in 2000, 2001, ourselves, along with the USDA and Cornell, got a fairly large grant to establish sort of honeybee genetic reserves, we'll call it, because of the fear that Africanized bees were moving up into the areas where queen production takes place in this country. And so we've continued selecting for stock that do well in our winters here, and that we don't use very much chemical control for the mites, and we don't use antibiotics to prevent bacterial diseases. So we've just been trying to select for better bees. And then the very last one here, this is kind of an example, is the breeding and propagation of Apis mellifera caucasica. This is a honeybee from the Caucasus Mountains, and it was brought in the 80s, but had not really been very well established before the Honeybee Act of 1922 came about. So this is a large black honeybee, really adapted for cold climates, very gentle, and there's an increasing demand for this kind of honeybee because, especially say hobbyists or people that don't need the colonies to be very strong all the time. Let me just say one slight digression, which is the preferred honeybee in the US for many years has been what are called Italian honeybees. They're yellow and they're very prolific. So if you feed them, they expand their brood nests and they get really big. And so that's what beekeepers, commercial beekeepers, have wanted in the past, because their main pollination sort of opportunity is with almonds, and almonds bloom in February, March in California, which is normally when the honeybees, say here in Pullman, would be hunkered down and waiting for summer to come along. So the beekeepers take bees down to California, feed them, they expand, the colonies get bigger, and the beekeepers are paid by the size of the colonies when they're in the almond. So that's why that particular subspecies or type of honeybee has been very popular with commercial beekeepers. Of course, if you're trying to keep your bees through the winter in Washington State, that's not the best trait you want. A bee that eats a lot of honey and gets strong. You want a bee that is very conservative of their winter stores and kind of hunkers down. And so these bees that are adapted for mountains and cold climates, as you might imagine, are fairly conservative of their winter stores, and those traits are beneficial if you're going to keep bees in cold climates. So that's when we brought that one back, and I'll explain more about that later. So the breeding program historically, we started, I had a master's student back in the late 90s, and we were selecting for a number of apicultural traits and kind of working out the genetics of that. And then we got that grant, the large grant, and we sampled over 2 years, 2001, I think, 2001 and 2002, from queen producers around the country. We basically set up a population mirroring the diversity that we had in commercial stocks. And that's what we've been selecting from until about 2009. Oh, by the way, this is our quarantine apiary. The USDA wanted us originally to go onto some kind of island. And I said, do you know where Pullman, Washington is? And in the end, they accepted that this place was an island in a sea of wheat. So as you might guess, there aren't a lot of beekeepers that are keeping bees in the wheat fields around here. And so this was a fairly isolated location as far as bees are concerned, and it's where we do a lot of our mating station at Smoot Hill. So we continue the selective breeding, and so that's always gone on. Every year we are selecting from among our own stocks. But then we began reaching out and getting new genetic diversity from some of these subspecies. So in the upper left panel over here, this is Sue Kobe, and this is in the country of Georgia, as is actually all three of these are in the country of Georgia, in the Caucasus Mountains, and we are collecting drones, or the males, from colonies, and then this semen will be brought into the US, or was brought in the US, and then we use instrumental insemination to inseminate queen bees. And Sue herself and Tim Lawrence historically have done a lot of work with Apis mellifera carnica, which is the honeybee of the Alps from Slovenia and Austria, places like that. Another dark bee adapted to cold climates. And this is Brandon's contribution, the ability to freeze the honeybee semen. This is actually in France. No, this is in Italy. We actually were collecting Italian semen because of the interest in the commercial beekeepers. So you put the semen in this little device here, and then the computer controls the rate of freezing. It's part of the process. Yeah, so what did we do with this old world germplasm material? We developed these Caucasian honeybees for reintroduction. So we initially started with a Carniolan virgin queen, a number of them, and then inseminated them with Caucasian semen. And then from those daughters that were obviously 50-50, we then inseminated those with Caucasian semen. And we kept repeating that. It's called back crossing until the line that we had was basically 95 or above pure Caucasian. And then that's what we've been releasing to industry. And the number 2 there, the New World Carnival and Breeding Program, that was what I told you that Sue Kobe had been sort of the mover and shaker in. And then finally, we've been bringing in the Italian genetic material in collaboration with a large queen producer in California, because as you could guess, being a university bee program, we don't have thousands and thousands of colonies that you would need to maintain all of these different genetic types. And so we've relied on beekeeper collaborators for the work on the Italian. So. This is Herrick Olsen. This is some of our early collections in Georgia. He's retired now. I say he's retired. He still grows a lot of cherries and pears in the Natchez area, but he was the largest beekeeper in Washington State, and his daughter happened to be in the State Department and work in the embassy there in Tbilisi, Georgia. So our initial collection of this I should tell you that the Caucasian honeybees, although they were maybe when I was a kid in the 60s, you could still buy what people called Caucasian honeybees, but they were really bees selected that were kind of black. So this was, we sort of were able to resurrect this subspecies or this strain for US beekeepers. And this was just a trip there. And there's Jackie Park Burris, and you can see these, I don't know, well, you can't, you see how these bees are yellow, nice and yellow, beautiful bees. And she was pretty excited. And so she produces 100,000 or so queens a year, and her and a number of other producers have been using some of this new genetics to increase the diversity of their own stocks. Yeah, I think I already told you this, that basically when we bring the semen back, they go into this apiary, and then we send the semen to the USDA, and it's only released after they find that it's not full of viruses. And then this is Jackie Parker, her husband, and this is, they've driven up from California to Pullman, and each one of these little nucleus beehives has an instrumentally inseminated breeder queen in it. And they will get, a certain number of them, like 5, and then there are a number of other queen producers, and every year they just send one person or, one team up, and we give it, and then they distribute it down there. This year I met them in Walla Walla. Oftentimes we meet halfway. That's a little bit harder for us. In this case, they wanted the person picking them up wanted to get some wine in Walla Walla. So they went up there for a day or so, and then we met them. And then the very last thing on this breeding program, these are both queen producers in California. They're both fairly large queen producers. And this is the USDA's animal plant, it's the National Animal Germplasm Program. So what you can't see is there are probably 150 of these giant liquid nitrogen tanks. This is a huge room. But in this tank 71, we have the honeybee semen that we've deposited in this National Animal Germplasm Program. The idea being that the USDA maintains a reserve of the genetics of all plants and animals of agricultural significance. So they have, semen from pigs, cows, chickens, sheep, oysters, all kinds of things here in this facility. And now they have honeybees. Yes, okay. The future goals, we're working on a way to basically come up with a biological materials license. This has been done sort of piecemeal with queen producers, where they pay for they pay for these queens. They buy these breeder queens from WSU, and then we use that to help keep the program going. But as you might imagine, this is fairly expensive, and we've been able to get grant funding or industry-based funding from commodities, from the honeybee point of view, the almond board, the California Bee Breeders Association. A number of these groups have donated money that have made these trips possible. But we would like to come up with a more stable system, and the biological materials license is a way to go along that. So we're working on that. As you know, Brandon and his students are trying to improve the methods for cryopreservation. There's a real interest to do this worldwide. There's one other laboratory in Germany that's been working on cryopreservation, but they haven't been using it in any practical sense or for breeding. But there are a number of countries, from their point of view, it's conservation, right? So the country has a limited amount of their indigenous sort of honeybee populations, and they want to preserve that diversity. So a group called Apimondia, which is kind of the World Beekeeping Congress, there's now an international effort to set up basically gene banks or these genetic repositories in different continents. So you can imagine, we here in the US, for us, we want to breed bees for our use, but we don't have a distinct conservation point of view because they're not native here. Whereas if you're on the island of Malta, they have their own indigenous honeybee subspecies, and a lot of people that are bringing in bees from Italy that are a different subspecies. So they're trying to preserve what they have before it's gone. And because of the way the European Union works, they do not restrict movement of bees across different countries in the EU, with the exception of Slovenia, which got an exemption because they only want their honeybee. And so that's increased the need to have some ability to save their material. Right, and maintain foundational stock for Caucasian, Carnival, and Italian honeybees. Those are there are about 24 different subspecies, and the three of most interest are those three right there. And we've been working with a fourth one called Pollinator, which is a honeybee native to the wild apple forest of the Tian Shan Mountains of Central Asia. And so this is a honeybee that we think might be a better apple pollinator. So, anyway, that's kind of where that's going. There's a queen, by the way, they don't come with their wings cut like that. We clip them only so we can make sure when we go in there the next time that was the queen we put in there. Okay. Are there any questions on? I feel like I'm just talking, but if you have questions on this, we could stop for a minute, and then I'll take the last 15 minutes to finish up.
We had a question about the Giant Hornet. If you want to take a just, what's the true nature of the threat from the Giant Hornets, and how fast can they invade a territory? So is panic warranted right at this point?
Kids never. No. Okay. The main, for example, South Korea has not had a major, public health disaster because of the Giant Hornet. So, it is very defensive of its nest, but most of these insects that have developed stinging as a defense mechanism, they're not particularly aggressive, out in the real world. So when they're out, getting nectar off of a flower or something like that, they're not looking to come over and sting you, because that increases the risk for them as well. So the main danger, I think, aside from people being, kind of afraid of them, is the devastation they could wreak on the beekeeping industry, and which of course is intimately tied to our food supply. So we really don't know. That's a really good question. I don't know how rapidly this one would spread. So we're always worked kind of the other way. people have brought Apis mellifera, the European honeybee, into the range of the giant hornet, right? So now in Japan or China or South Korea, the main honeybee of commerce, the honeybee that they make money with, is the European honeybee, not their native Apis cerana honeybee that has the defense against it. So the answer for that was I was in Thailand talking to someone from Nepal, and Nepal was one of the last countries that tried to keep out the Western honeybee, and did that at that time by royal decree. But beekeepers managed to get Apis mellifera, and the beekeeper's story to my colleague was, I kept Apis cerana for 10 years and rode a bicycle, and now I have Apis mellifera and I drive a truck. So they can make a lot more money with Apis mellifera, and that's kind of driven things a little bit. So I don't think, not that we ever needed to be afraid of Africanized bees either, as a public health menace. So I would just say be vigilant, be cautious, teach your children not to throw rocks at hives or stinging insect nests they don't know. It's never a good idea. And I think we just have to see.
Okay.
Yeah. So
I have another question that says,
yes,
what was the fungi that is used in the hives first discovered? And maybe you'll get to that with the rest of your presentation.
Sure. Yeah, I can go on with that. Well, remember there are two things. One is the fungi that actually attacks and kills them. And that fungus has been known for a while. There are some registered products for using it for things like termites or carpenter ants. Those would be different strains of the fungus, but they're known to be safe. I think that they even use them in kitchens and things like that. And so that's, and there was a good bit of work on the honeybee, maybe back in the 90s or 2000s, but it was deemed to be not practical. We've been working with Fungi perfecti, and they grow, they can grow tens of thousands of kilos of fungus a week. And so they don't believe that it's going to be a difficult thing to grow it up in quantities suitable for the use by beekeepers. The other one, the properties of these fungi, these polypore mushrooms, to contain antivirals has been known a long time, and they're being explored a lot in human medicine. And in fact, Paul, who contacted us about the sort of the initial testing of this, first got results back after 9/11. The NIH allowed natural products to be screened for antiviral properties. It was cited that there may be domestic, terrorism that used bioweapons, and so they kind of expanded what they would look at from the, I'll just say, from the general public or the scientific community. And Fungi perfecti had a number of their polypore cultures, because they have hundreds of different fungi in culture. They had those analyzed by NIH, and a number of them came back with really high antiviral activity in like human, papillomavirus and a number of SARS, a number of viruses that were being tested. So that's a separate track and kind of being explored. And it was, we're sort of the first to look at this with honeybees. So it's been in the last decade or so for the polypore mushroom stuff. So let me try to zoom through this without taking up too much time. So here's a Varroa mite and a honeybee. So you can see it'd be like having, a pretty big saucer or maybe a small plate of an animal attached to your back or wherever they go. And they spread viruses quite a bit. These are the types of fungi we're talking about, polypore mushrooms. They've been used for a long time. Some of these mushrooms, their fibers are dissociated in in lye water, and they can be felted and used to make clothing, or to be used to hold fire, which is what they were in the, in the Stone Age, I guess, in Europe, or whenever, before the Middle Ages. But when you're walking around trying to keep fire, they would use some of these fibers, and they would smolder all day long. So I would imagine one person's job in the clan would be to keep the fire going, till they got to the next place. Okay, and it's used for packaging, and there are innovative uses. Here's an ant with a different kind of fungus. It's now fruiting out of its body, something coming out of the ant's head. But this is the Metarysm. So this is the one that we're talking about that directly attacks the mite. It's an entomopathogenic fungus. It's native to the US. You can find it probably if you go outside and dig in the soil in your yard, you've got it. So, the insect crawls around through the ground, has a little scratch on it, and then this fungus can attack it and kill it, and then the insect becomes a big fruiting body. So this, here, I'll use my sign. I'm pointing at it with my finger. It doesn't help. So all these stuff caked on it are billions of spores. Here's a tick with spores all over it, and then they, can be infected for the next thing. And we've done a lot of experiments with this, and it doesn't seem to hurt honeybees, which is always a good thing. Okay, we did initial experiments where we just put little petri dishes full of this fungus in beehives, and then counted mite fall. And let's see, and what you can see, forget this preconidial thing. This was a different strain that we were trying that does not make spores. But the normal one that makes spores, you can see here on day 5 and 7 in this hive, there was significantly more mite fall than in the controls, right? So these guys were getting nothing. These guys got the little petri. There are more mites falling. This is kind of, the range of number of days where the mites fall. And the other exciting thing that, let's see if I have a picture of this. I may not have done it. Darn it. What Jennifer did was when the mites would fall off the bees down into the bottom of the hive, she would then take all of those mites and plate them out on a agar plate, and the ones that had died from the fungus would, of course, grow more fungus. And so then she would raise the next generation of fungus from those. So basically selecting for virulence every generation. And that led to a number of things. One of them being that the fungus is now much more virulent toward mites. It's much more able to deal with its higher temperatures. And I'll kind of skip through. When the spore touches a mite, it sends out a little thing called an Apresarium. You might think of it as a little spear on a string. And so here it is, and you see the length of this. So the length of this is related to the infectivity of the spore. And so here's how they used to be, and here they are now in this lower panels, how they are after a number of generations of selection. So basically these spores are much more infective than these spores through selection. So it's kind of cool, this regular animal breeding or, plant breeding, but, not really, a molecular biology. It's just standard old breeding, like breeding from the cow that gave you the most milk. So that's it's been kind of fun. There's also some evidence with just individual bees. Sorry, those were in, dadgummit. Those were in cage trials, and it increased the longevity of bees, a little bit. And you can see the early years, what you can see here is sort of the area under the green lines. The orange lines are the treatment. The area under the green lines are the percent mycosis, the mites that are dying from fungus. And you can see it's increasing. So the selectivity of multiple generations has led to a more virulent fungus. Same thing here, it's just 1 year difference. Yeah. And percent of spores at 35 degrees, the same idea that you can select from strains that germinate a lot better at higher temperatures, like in a beehive. And then the next step was to figure out how to apply this in the beehive. Giving beekeepers, petri dishes is not going to be the answer. So I tried using felt strips impregnated with it. And the latest thing that we're doing are growing them on grain and putting them in bags. And you put that bag of infected grain into the hive. This is just showing you that you can get control or mortality of the mite. That's similar to oxalic acid, which is a commercial thing that we use. So here you get 60% mortality with 1.5% oxalic acid, and at a higher concentration of spores, you get as good or better control using the fungus. At low concentrations of spores, not as good. The cage studies have really pretty much been done, and this field study using oxalic acid and Metarysm has been done. Really, the next steps now in the work that Jennifer and Nick are doing has been to try to develop sort of a practical method for large quantities, and we're working toward getting, for WSU, is working toward patenting this. Hey, Kara, I realize it's 1 o'clock. These are some field experiments using the stuff. I don't know. Should I stop now? I think we're time limited here. These are just some field experiments using Varroa mites and the virus and these compounds. The idea being really that when honeybee colonies get really infected, they become what's called a mite bomb, and it spreads throughout the apiary into other beekeepers. So when beekeepers all go to California, it only takes a few, well, to say bad apples, for everybody to get really infected with a lot of mites, and then they spread the viruses around. So the idea being that if we can control the viruses with those extracts, we might be able to, the mite may not be such a big problem. All right, I think maybe I should, I was just gonna talk about that, and then we could just go to questions.
Sure. People are signing off right now, but before everybody signs off, I do want to give a little plug for our next month webinar, which is going to be talking about ag labor issues with April Clayton and Rosella Mosby. So don't miss that. You can sign up on that at our ag forestry website. But we do have a couple more questions that came in, Dr. Sheppard. One of them was, could the giant hornet survive in the climate of Eastern Washington?
I believe the short answer to that is certainly, yeah. So they don't overwinter, as a colony. Just individuals, hide under a piece of bark or a rock or in the eaves of your house or under a piece of tarp that's out in the ground, and they become inactive, and they can definitely survive in Eastern Washington.
Okay. How about in Thurston County, they have a 30% overwintering survival rate. Does the Caucasian bee strain have a higher survival rate? And are they susceptible to mites?
Yeah, a good question. That's tough to say. I would say this Thurston County is around Olympia and everything. The survival rate of honeybees, has a lot to do with the care they receive. I'll just say not so much, the genetics. I mean, everything being equal, a Caucasian or a Carniolan colony in a cold climate would be more pre-adapted for being able to survive it, right? So, for example, say here in Pullman, we have a hard winter. Okay, no big deal. Italians, Caucasians, Carniolans are all doing fine. And then we get a couple of weeks of really warm weather, which we sometimes do it. Hey, spring is here, spring is here. The Caucasian and Carniolan honeybees I'm kind of anthropomorphizing here. They're not fooled by that. They just kind of hunker down and keep doing what they're doing. Maybe it's spring and maybe it's not. Whereas the Italian honeybees, oh, spring is here. And so they begin really expanding their brood production and, starting to grow fast, which is what they do. And then if we get another few weeks of really cold weather, which we sometimes do, they're left out in the lurch and may die because they have to withdraw from where they've expanded to stay warm enough with the cluster, and they sometimes get off their food more easily because they've expanded, and eaten a lot of food to start making baby bees, and then those baby bees, of course, freeze. So in general, they're better for cold climates, but Thurston, it's not a super cold area, but they overwinter as a smaller cluster, and they're more conservative of their food. So if those are the things involved in the 70% that aren't making it, then it would be better. But oftentimes there are other factors that lead to why colonies don't make it through the winter. Usually they're not in great shape going to winter. And then, if you don't have the bees really ready to make it through the winter by the end of fall, there's very little you can do to save them.
Another question is the fungal treatment available to hobby beekeepers at this time?
I guess the short answer is no, but we're working as hard as we can to make this available. And I believe that, hobby beekeepers would be one of the big users of it. Hobbyists are non-commercial beekeepers are oftentimes able to spend a little more time with their bees and do things that take a little more labor. So, for example, there's one product available now called Hop Guard. It's made from hop beta acids, and it works well, if you don't have a lot of brood. So if you can either remove brood and put it in there, or treat them at certain times of the year, you can get pretty good control with that. And that's not so easily used by commercial beekeepers. So it's not available now, but we're doing what we can to get it available to everyone.
Okay, I have another one for you. So the fungal patches and the fungal grain, that is not yet available commercially.
I'm sorry, the fungal, what were those words?
The patches, the felt patches, and the grain.
Right, no, man, they're not available commercially.
Okay,
no.
So where can they learn more about the cold storage studies and any effective means of using it for small scale operations?
Oh, that's a great question. There is, Brandon has put out a booklet by, I believe, if people look up, you can either write Brandon Hopkins or go to Project Apis M, which is a nonprofit out of California. And they have produced a book or a booklet that Brandon wrote on indoor storage. So that would be a great place to go. Or, just write Brandon here. You could probably send you a PDF copy. So Brandon Hopkins at, WSU.edu. I think it's b.hopkins at WSU.edu if you want to write him. Otherwise, go to Project Apis M online. And I believe this PDF book will be, it's free.
And one more question. Any idea what percentage of Washington ag crops depend on honeybee pollination?
Wow, that's a great question. I would say that, I mean, if you say that one of our major, although they're called minor crops, right? Apples, I know we use about 250,000 pollination sets for apples. So I don't know the percentage of crops, but certainly some of our major crops, rely on honeybee pollination. And obviously not wheat or lentils or anything like that out here, but for tree fruits and for small fruits, a lot of vegetable seed production over on the west side, onion seed production, cranberries, they all require or benefit from honeybee pollination.
Great. Dr. Sheppard, thank you so much for your time today. It was an absolute pleasure and I learned so much. I thank you very much from the bottom of my heart and from Ag Forestry as well. And we will be recording this and putting it up on our website if that's all right with you so that people can take a look at it later as well.
Sure. Thank you, Cara.
Thank you. Have a great day.
Okay. You too.
Thank you.
Bye.